from the purpose of study the actual function of the gene, it is very important to study the actual function of gene at a molecular level. Okay, so that's why many scientists have done this research on the functional genomics, through which they have not only studied the DNA structure and all DNA, there's a DNA sequence, but they actually study the function of the gene by doing various experiments. Okay, starting from the discovery of DNA, so many scientists have shown interest inside the study the molecular level or the character or what we call the function of that DNA sequence or gene. Okay, so that's why they have developed so many techniques to identify the function. Okay, so ज्यावेळेस सायंटिस्ट ने or actually वॉटसन आणि क्रिकने ज्यावेळेस डबल हेलिक्स मॉडेल दिले आपल्या डीएनएचं, त्यानंतर सगळ्या सायन्सने त्याच्यामध्ये इंटरेस्ट दाखवला. ओके, की त्या डीएनएचं ऍक्च्युअल फंक्शन काय शकतो. ओके, स्टार्टिंग फ्रॉम ग्रिफिथ एक्सपेरिमेंट, त्या ग्रिफिथच्या त्या एक्सपेरिमेंट पासून माईसच्या तर डीएनए ट्रान्सफॉर्मेशन किंवा ऍक्च्युअली जे study करायचं. Okay, the whole genome we need to study. Okay, in the process of development of this genomics, the genome study of an ideal plant or individual plants, it is needed. Okay, गरज होती. त्यानंतर मग मेनी सायंटिस्ट, डेव्हलपमेंटचे सायंटिस्ट होते, त्या प्रत्येक सायंटिस्टने जिनोम इंडिव्हिज्युअल प्लांटच्या जिनोमवर स्टडी करण्यास स्टार्ट केलं. ओके, so our topic in our syllabus is to study the research project or a genome project on a Arabdiopsis E. coli, okay, and human genome project. Okay, so why they choose this organism? It is mainly because it is a model organism. Okay, in every category, in the plants they choose Arabdiopsis thaliana. Okay, because it is a model organism. Then after in the microorganism, that is a bacteria, they choose E. coli. In the fungi class they choose yeast. Okay, then in the insect they choose Drosophila. Okay, Xenophatis ilingus is the nematode. Okay, they choose this as a model organism. Okay, from the every family or organisms family point of view, they have chosen the model organism. Okay, from that. Okay, but why Arabdiopsis they have chosen? Because it is a model plant. But why it is a model plant? It has a convenience and abundance. Okay, so the abundance, that means it's available to easily. Okay, so that's why they choose the Arabdiopsis. It is relatively smaller and simpler genome. Okay, as compared to the other organism, it has a relatively smaller and simpler genome. Okay, so that's why they have chosen this Arabdiopsis as a model plant to study the genomics. Okay, because at the starting point, the scientists needed to have a very smaller segment or smaller amount of DNA. Okay, sorry, 1 minute, please. Okay, sorry. So they have chosen because we need at the starting point we need a smaller amount of DNA. Okay, a smaller amount of genome sequence that we need to study. Okay, so that's why they have chosen this Arabdiopsis. Okay, as a model plant. Then after susceptible to tDNA insertions. Okay, so while we are preparing the genomic library to study the genomics of that plant, we need to insert that segment of DNA inside a host cell. Okay, to prepare a clones and prepare a genomic library, we need to prepare the tDNA insert. Okay, so that's why we need to have the genome segment that is susceptible to tDNA insertion. Okay. So actually, you know what is the tDNA insertion? That in Arabdiopsis, the segment tDNA, okay, transfer DNA. Okay. So the basic similarity to other crops, as it is a model plant already, so it has a basic similarity to the other crops. Okay, it is not very similar, but it has showing some basic similarity to the other plants. Okay. It shows the similarity to the angiosperm, gymnosperm, all the plants. Okay. So low amount of repetitive DNA. It has very low amount of because it has a very small segment of DNA or small and simpler genome. That's why it has also a low amount of repetitive DNA. Okay. So as we seen in the microorganism, that it has a low or a smaller segment of DNA or smaller segment of so that's why there is low amount of repetitive DNA inside the aerodiosis, and its genome size is in the nuclear, it is about 125 megabase pair, and so that's why it has smaller genome size, larger number of offspring and short generation times. Okay, it has a larger number of offspring. That is means it can generate a larger number of offspring in the short time. That's the advantage of that plant. And then after this small plant belongs to the family Cruciferaceae. Okay, we need to study generally the program of this genomic research project. Okay, only. So how does it completed and what are the outcome of that project? Okay, so that's the main purpose of our study. So starting from 1983, the first genetic map was published. Okay, by the scientists. And after the publication of RFLP maps are generally started in 1988 to 89. Then after the project of multinational coordinated aerodrosis Thaliana genome, it started in 1990. Okay, so basically at that time many scientists from the uh, so many countries work on a single project. Okay, nowadays also it is happen. Many scientists also work on a single crop. Okay, so they coordinate between themselves. Okay, so they generally having the purpose to study the crop. Okay, so generally in the case of rice also and wheat, our country also contributed some part. Okay, of the sequencing in the case of rice. Okay, so so that's a part of the research project that scientists from many countries work on that project. Okay, so it happens not only in the life science, it happens also in the physics and other science research project. Okay, so in 1991, first YSC, that is yeast artificial chromosome libraries were prepared. 1995 to 96. So okay, that time the genomic libraries were prepared by using different vectors. Okay. So at that time, the YSC, that is yeast artificial chromosome and bacterial artificial chromosome, were having the largest genome insertion site. Okay, and carry largest amount of DNA. That's why they were used as a cloning vector. Okay, then prepare the Arabdiopsis genome. Okay, okay. In 1996, the Genome Initiative organized and started sequencing. Okay, so the sequencing of the genome. Firstly, they prepare the genome library. Okay, then after they have started to sequencing the genome. Okay, then after in 1998, the physical up till now you have understand all this. Okay, तर मराठीमध्ये सांगू का ओके, प्रोसेस कशी झाली तुम्हाला मराठीमध्ये सांगतो. ओके, स्टार्टिंग इन 1990 मध्ये एक मल्टीनॅशनल कोऑर्डिनेटेड अरॅब्डियोसिस प्रोजेक्ट होता. ओके, जिनोम प्रोजेक्ट. त्यांनी पहिले जिनोमिक लायब्ररी जे असते त्या क्रॉपची. ओके, आता जिनोमिक लायब्ररी प्रिपरेशन तुम्हाला माहिती आहे की क्लोनिंग वेक्टरच्या माध्यमातून आपण तो जिनोमिक लायब्ररी बनवत असतो. ओके, सो तिथे वायएस ईट आर्टिफिशियल क्रोमोझोमच्या लायब्ररीज बनवल्या. ओके, सो त्याच्या त्या यूज का केल्या गेल्या कारण त्या टायमिंगला त्या हे जे वेक्टर होते ईस्ट आर्टिफिशियल क्रोमोझोम आणि बॅक्टेरियल आर्टिफिशियल क्रोमोझोम हे वेक्टर लार्जेस्ट अमाऊंट ऑफ डीएनए कॅरी करू शकत होते. ओके, वेक्टरचं काम आहे डीएनए कॅरी करणे. सो जिनोमिक लायब्ररी बनवण्यासाठी या वेक्टरचा त्यावेळेस यूज केला गेला आणि 1996 पर्यंत ह्या जिनोमिक लायब्ररी प्रिपेअर झाल्या. त्यानंतर ते ऍक्च्युअल सिक्वेन्सिंगचा पार्ट जो आहे तो 1996 नंतर स्टार्ट झाला. ओके, सो त्या टायमिंगला ज्या मेथड होत्या मेनली सांगर्स मेथड, शॉर्टगन मेथड. ओके, त्या मेथडचा यूज करून तिथे त्यांनी सिक्वेन्सिंग स्टार्ट केलं. आता सिक्वेन्सिंगला 2 वर्ष लागली. त्यानंतर 1998 मध्ये त्यांनी फिजिकल मॅप ऍक्च्युअल स्टार्ट केले. ओके, बनवायला क्रोमोझोमचे. आता फिजिकल मॅप्स कसे बनवले तर जे क्रोमोझोम असतात फिजिकल मॅपिंग. ओके, त्याची जिनोमिक लायब्ररी आपण बनवले त्याला रेस्ट्रिक्शन एन्झाईमच्या थ्रू क्लिवेज करून त्याचं ऍक्च्युअल रेस्ट्रिक्शन साईट किंवा त्याची साईट तुम्हाला माहित असतं. त्यातलं ओव्हरलॅपिंग सेगमेंट फक्त आपण कट करून जो कंटिन्युअस सेगमेंट आहे तो मॅप करतो. ओके, ते होता रेस्ट्रिक्शन मॅप्स और फिजिकल मॅप्स तयार. ओके, फिजिकल मॅप त्यांनी 1998 मध्ये तयार केले आणि त्यानंतर त्यांनी क्रोमोझोम वाईज जे सिक्वेन्स आहेत त्याचं अनालिसिस स्टार्ट केलं. तर 2 नंबर आणि 4 नंबर क्रोमोझोम स्टार्ट केलं 1999 मध्ये. 2000 मध्ये 135 नंबरचं त्यांनी क्रोमोझोम अनालिसिस केला. ओके, सो होल सिक्वेन्स अनालिसिस वॉज कम्प्लिटेड इन द इयर 2000. ओके, ओके, ते प्रोसेस पुन्हा कंटिन्यू राहिली पुढे. पण त्यामध्ये 2000 पर्यंत जवळपास बऱ्यापैकी 99% त्यांचा जिनोम सिक्वेन्स झालेला होता. ओके, सो ऍक्च्युअल तो जो रिलीज केला गेला डाटा अरॅब्डियोसिसचा तो 2002 मध्ये केला. ओके, प्रोसेस समजली का? Okay, then after analysis of the genome sequence of the flowering finder. सो त्या रिपोर्ट मध्ये त्यांनी काय दिले होते काही हायलाइटेड पॉइंट्स होते. ओके, तुझा रिपोर्ट रिलीज झाला ज्यावेळेस जिनोम सिक्वेन्सिंग कम्प्लीट झालं 2002 मध्ये. सो दिस रिपोर्ट इन्क्लुड्स completed Arabdiopsis genome sequence. Okay, then after annotation of predicted genes. Annotation is nothing but assigning the function and name to the gene. Okay, सो annotation काय केलं त्यांनी की जे जीन्स होते प्रिडिक्टेड त्यांचे फंक्शन किंवा त्यांचे जे काही ऑन द बेसिस ऑफ सिमिलरिटी दे हॅव असाइन्ड द फंक्शन अँड द स्ट्रक्चर ऑफ दॅट जीन. ओके, सो त्याचा सिक्वेन्स होता त्याच्या बेसिसवर त्याने अनोटेशन केलंय. त्यानंतर असाइनमेंट ऑफ फंक्शनल कॅटेगरीज, chromosomal dynamics and architectures, distribution of transposable elements. Okay, so it is also important parts, transposable elements and other repeats, extent of lateral genes transfer from organelle, comparison of the genome sequence and the structure to that of other Arabdiopsis accession and the plant species. Okay. सो सिक्वेन्सिंग स्ट्रॅटेजी मध्ये त्यांनी ह्याच्या स्ट्रॅटेजीज होत्या क्लोन बाय क्लोन सिक्वेन्सिंग. दॅट मीन्स दे हॅव प्रिपेअर अ जिनोमिक लायब्ररी, देन दे हॅव sequence the clone by clone. Okay, then after hierarchical shotgun sequencing, map based shotgun sequencing. Okay, these are the strategies they have used. Okay, So it generally includes map construction, then after clone selection, then after subclone library construction, random shotgun phase, directed finishing phase. So these are all the phase they have completed. Okay, in that we need to study only the phases. Okay, we need not to go in detail because it's a whole genome sequencing project. It's very huge. Okay, in that we need to study only the strategies they have used and okay what are the steps inside that. So in that the random shotgun phase is generally nothing but a sequencing. Okay, then directed finishing phase is nothing but a annotating the annotation of the sequence. Okay, that means the give the predicted function to the gene. Okay, then after sequence authentication is nothing but authenticating this finally it means to authorize. Okay, it is final step to authorize the all the information. Okay, okay, those are the steps in that. Firstly, primary substrates. So they have prepared the large insert inside the BSC, okay, TAC libraries, the physical map of the genome of accession. Then after that, they prepare the physical map of the genome. Selected minimally overlapping 1569 BSC, TSC, and okay, and P1 clones. So they have average inserts about 100 kb. Okay, in that they have assembled 10 quantics. Quantics is nothing but the segment of that chromosome. Okay, or a segment of DNA. Okay, so it has assembled 10 quantities represent minimum killing path. Okay, so these clones are selected for shotgun sequencing after that. So why they have chosen the minimally overlapping? Okay, because they need not because the cost at that time to sequence the DNA is very high. Okay, so that's why they have chosen the minimally overlapping, okay, segment of DNA. Then after they have to clone the sorry, sequence the clones. Okay, to link the region not covered. Then they also subclone this into the plasmid or EM13 vectors. Okay, after doing the physical mappings, they have generally inserted the segment of DNA after physical mapping into the M13 patch, okay, and plasmid vectors. It is a method of subcloning. Okay, then after they have generated the data that is so after subcloning they have generated the data by using the method of sequencing. Okay, then after they computationally assemble those segments. Okay, so त्यामध्ये काय होतं की पहिले क्लोनिंग केलं. ओके, लार्ज सेगमेंटचं नंतर फिजिकल मॅपिंगच्या मेथडच्या थ्रू आपण त्याचं रिस्ट्रिक्शन एन्झाईमनी केलं सेगमेंटला. ओके, सो आता तुम्हाला माहिती आहे की त्याच्या सेगमेंट आहेत त्याच्या साईज किती असणार आहे. ओके, कारण रेस्ट्रिक्शन मॅपिंग या मेथडच्या थ्रू तुम्ही तिथे त्या पर्टिक्युलर साईजचा तिथं फ्रॅगमेंट तो क्लिव केलेला आहे. ओके, सो त्याला तुम्ही पुन्हा एकदा सब क्लोन केलंय M13 आणि प्लाझ्मिड DNA मध्ये, प्लाझ्मिड वेक्टर मध्ये. त्याची सब क्लोनिंग करून त्याला पुन्हा तुम्ही आता त्या सेगमेंटला त्याचं सिक्वेन्सिंग करताय आणि सिक्वेन्सिंग केल्यानंतर तिथे जो डाटा येतो त्याला कम्प्युटेशनल तुम्ही असेंबल करताय. ओके, असेंबली केल्यानंतर तुम्ही तिथे त्याला अॅनोटेशन आणि व्हेरियस मेथडच्या थ्रू तिथे तुम्ही त्याला ऑथराइज करताय शेवटी. ओके, व्हेरीफाय करताय. ओके, सो आउटकम काय होते त्या प्रोजेक्टचे? सो मेनली दिस अबाउट अप्रॉक्सिमेटली base pair are sequence. Okay, this unsequenced centromeric and ribosomal DNA repeats region measure about roughly 10 megabase pair. Okay, ते काही जे सेंट्रोमेरिक रिजन आहेत किंवा डीएनए रिपीट्स आहेत रायबोसोमल त्यांचे नियर 10 मेगाबेस पेअर असतील असं त्या रिपोर्ट मध्ये दाखवलंय. त्यानंतर 25,498 जीनचं त्यांनी फंक्शन प्रेडिक्ट केलंय. ओके, ओके, आता ही जी माहिती होती ते डाटाबेस च्या माध्यमातून स्टोअर केली. प्रत्येक क्रोमोझोमचा त्यांनी स्टडी केला होता. तर त्या म्हणजे क्रोमोझोम ऍक्च्युली अरॅब्डोसिस मध्ये 5 क्रोमोझोम आहे फक्त. तर त्या 5 क्रोमोझोमचा त्यांनी स्टडी केलाय. ओके, what are the outcomes of the sequencing project? It's mainly to characterize the coding region, genome organization and duplication, comparative analysis of Arabdiopsis. So we can also do comparative analysis. Okay, so because if ज्यावेळेस तुम्ही सिक्वेन्स स्टडी करता प्रत्येक ऑर्गॅनिझमचं त्यावेळेस तुम्ही कंपॅरेटिव्ह जिनोमिक्सचा पण स्टडी करता. ओके, so by doing blast and similarity, okay, analysis, you can study the comparative genomics also. So you also they also study the transposable elements, RDNA, telomere, and centromeres. Okay, all these are the repeating sequence, and it also has a very important role inside this. Okay,